We've just explored how the hormonal shifts around menstruation can influence aggression and irritability. That's a fascinating example of hormones impacting behavior, but it also touches on the broader theme that our brains aren't static monuments. We know experience can tweak synapses, prune dendrites, and even spark new neuron growth in adults. But here's the kicker. These changes aren't just minor tune-ups, they can be so significant they actually alter the physical size of different brain regions. It's like the brain is sculpting itself based on life's demands. Let's look at some concrete examples. The hippocampus, our memory hub, especially for spatial maps, doesn't just stay put. Give postmenopausal women estrogen therapy, and bam, their hippocampus gets bigger, likely due to more dendritic branches and maybe even more neurons. Flip the coin, chronic depression, severe pain, or conditions like Cushing's syndrome where glucocorticoid levels are sky high cause the hippocampus to shrink. This shrinking isn't just a number game, it's linked to real cognitive problems, memory loss. Think of it as the brain's memory palace getting either renovated or falling into disrepair under stress. Similarly, the amygdala, our fear center, tends to enlarge in people with PTSD. It makes sense, right? A brain wired for hypervigilance after trauma would physically reflect that state. Now, the exact mechanism, are we talking more neurons or just denser connections, is still a bit murky in many cases, but the effect is clear. Size matters and experience dictates the size. Now for a really cool story that brings this size change idea to life. Remember the London taxi drivers? They have to memorize the entire city map, the knowledge, considered one of the toughest tests globally. Researchers looked at their brains, specifically the posterior hippocampus, which is key for spatial memory. Guess what? Compared to regular Londoners, the cabbies had a noticeably larger posterior hippocampus. But was it innate, or did the driving make them bigger? A follow-up study nailed it. They scanned people before and after they went through the grueling multi-year process of learning the knowledge and passing the test. And the result? For those who successfully completed the training, their hippocampus grew during that period. It's like the brain physically expanded to accommodate the massive amount of spatial information being processed and stored. This isn't just a lab rat finding. It shows how intense, real-world experience can literally reshape brain structure in a relatively short time frame, months, not years. Beyond just changing the size of brain regions, experience dives deep into the molecular machinery. Think about receptors, the docking stations for neurotransmitters and hormones. Experience can change how many of these docking stations are present. Same goes for ion channels, the gates controlling electrical signals across neurons. And get this, experience can even flip the on-off switches on genes within brain cells, altering which proteins are made. It's like the brain has its own epigenetic control panel, constantly being tweaked by what's happening outside. Look at chronic stress again. In the nucleus accumbens, a key reward area, stress depletes dopamine. This leads rats to become socially subordinate and humans towards depression. On the flip side, winning a fight boosts testosterone receptor levels in reward areas, making the win feel even better. It's a powerful feedback loop. Even parasites like Toxoplasma gondii can hijack the brain, subtly changing behavior in rats to make them less fearful of cats, and potentially influencing human behavior too. The takeaway? Almost anything measurable in the nervous system is malleable. And crucially, often these changes are reversible if the environment changes. Your brain is incredibly adaptable. So discovering that adult brains can grow new neurons and rewire themselves, that was a big deal, overturning old ideas. It's exciting, and honestly, it feels optimistic. Books with titles like The Brain That Changes Itself or Rewire Your Brain tap into this hope, suggesting we can literally train our minds to be better. It's got that can-do spirit. But let's not get carried away. First off, plasticity isn't inherently good or bad, it's neutral. It's amazing how blind people's auditory cortex takes over visual processing, or how a cabbie's hippocampus expands. But equally, it's devastating when trauma enlarges the amygdala and shrinks the hippocampus in PTSD. More motor cortex for finger dexterity is great for a surgeon, maybe not so great for society overall if everyone becomes a safe cracker. Second, plasticity has limits. If it were limitless, grievously injured brains would just heal themselves perfectly. They don't. And third, practice doesn't guarantee mastery. Malcolm Gladwell talks about the 10,000-hour rule for expertise. But doing some 10,000 hours doesn't automatically make you a Yo-Yo Ma or LeBron James. There's a limit to how much plasticity can achieve, and innate factors play a role too. Okay, so we probably won't be spraying gene therapies up people's noses to make them more empathetic anytime soon. The direct application for complex traits is far off. But what's the real value of understanding neuroplasticity, especially in the context of psychology? I think it's largely psychological. It gives us a tangible scientific backing for the idea that people change. Think back to the PTSD hippocampus study. The author pointed out how ridiculous it was that some legislators needed brain scans to believe veterans were genuinely suffering. Neuroplasticity provides that kind of concrete evidence. It makes the functional malleability of the brain less abstract. When we see massive societal shifts, like the Arab Spring, or individual transformations, like Rosa Parks becoming a catalyst, or Mandela evolving from prisoner to statesman, neuroplasticity suggests that underlying these changes were actual rewiring in the brain. A different world creates a different worldview, and that translates into a different brain. Making this neurobiology seem real helps us grasp that such profound changes aren't just wishful thinking, they have a biological basis, making future transformations seem possible. Alright, let's shift gears slightly. We've talked about how the adult brain changes, but what about development? Specifically, adolescence. Does the brain operate differently then, leading to that unique blend of behaviors we associate with teens? Absolutely. One major fact dominates this discussion. Unlike the old dogma that brains are mostly wired up by age 2, the reality is much slower. The final brain region to fully mature in terms of synapse count, myelination, and metabolic activity is the frontal cortex. It doesn't go fully online until the mid-20s. This single fact has huge implications. First implication, no part of the adult brain is more shaped by the adolescent years than the frontal cortex. Second, nothing about adolescence makes sense without understanding this delayed maturation. Imagine this scenario. During adolescence, the limbic system, autonomic nervous system, and endocrine systems are all firing on all cylinders, pumping out hormones, driving emotions, and pushing for action. Meanwhile, the frontal cortex, the part responsible for planning, impulse control, and rational decision-making, is still figuring things out, still developing. You've got this powerful engine running, but the brakes and steering wheel are still being installed. This explains why adolescence can be so frustrating, brilliant, foolish, impulsive, inspiring, destructive, selfless, selfish, impossible, and world-changing all at once. Think about the peak periods for major life events: leaving home, starting relationships outside the group, inventing new art forms, committing crimes, becoming addicted, dedicating lives to causes. It's a time of maximal risk-taking, novelty-seeking, and peer influence. And according to this chapter, it's all fundamentally rooted in that still developing frontal cortex. Now a provocative question. Is adolescence even a real biological stage, or is it just a construct of modern Western culture? The argument goes like this. Better nutrition and healthcare meant puberty started earlier, while education and economic structures pushed marriage and childbearing to later ages. Boom! A gap appeared between biological maturity and social expectations. Adolescence was born. But the author pushes back, arguing neurobiology suggests adolescence is for real. The adolescent brain isn't just a half-baked adult or a child left in the fridge too long. Interestingly, many traditional cultures do recognize adolescence as a distinct phase, granting some adult rights but not full responsibility. What the West did invent, however, is the longest period of adolescence. Also, the intense intergenerational conflict often associated with adolescence seems more characteristic of individualistic cultures. Collectivist cultures might experience less of that specific friction. And finally, it's worth noting that not every teenager goes through a stormy period of acne of the psyche. So, if the frontal cortex matures last, does it just keep adding more neurons and synapses until it's done? Nope. Quite the opposite. During adolescence, the volume of gray matter, which reflects the density of neurons and dendrites, actually decreases. How can that be? It's thanks to a clever evolutionary strategy called neural Darwinism or pruning. The fetal brain initially overproduces neurons. Then there's a massive competition. Neurons that successfully migrate to the right spot and form strong connections survive. The ones that don't make the cut undergo programmed cell death, their components recycled. This results in leaner, more efficient circuits. Less is more. The same pruning happens in the frontal cortex during adolescence. By the start of puberty, the frontal cortex has more gray matter than in adults. Over the next decade, less effective connections are trimmed away. Older parts of the frontal cortex mature first. The newer, more advanced dorsolateral prefrontal cortex, DLPFC, crucial for complex thinking, starts losing gray matter later. A landmark study showed that kids who took longer to pack on that initial gray matter before pruning began ended up with higher IQs as adults. So maturation here is about efficiency, not just quantity. This focus on efficiency, not just size, is key. It often trips people up when interpreting neuroimaging studies comparing teens and adults. A common finding is that adults show more frontal cortex activation when performing complex tasks, suggesting better executive control. But flip the script, find a task where teens manage to perform at adult levels of control. In those situations, teens often show more frontal activation than adults. Why? Because achieving the same level of regulation requires more effort in their less optimized, still pruning brain. Think about detecting irony. Teens aren't as good at it, and when they try, they heavily activate the DMPFC, a core executive region. Adults, who are better at it, show more activation in the fusiform face area, involved in visual processing. What does this mean? For adults, spotting irony is partly a visual cue, almost automatic. For teens, it's a heavier lift for their frontal cortex. Their brain is working harder to achieve the same result. While gray matter is being pruned, what about white matter? White matter consists of axons wrapped in myelin, the fatty insulation that speeds up nerve impulses. Unlike the pruning pattern of gray matter, myelination in the frontal cortex steadily increases throughout adolescence. This is crucial because myelin allows neurons to communicate faster and more reliably. As myelination progresses, different parts of the frontal cortex become better connected, their activities become more synchronized, and the whole region starts functioning more like a cohesive unit. This really drives home a point that's easy to forget when studying neuroscience. The brain isn't just a collection of independent regions like cortex or hippocampus journals. It's about circuits and connectivity. Growing myelin during adolescence is literally about strengthening the wiring between different parts of the frontal cortex, making the network more efficient. Adding another layer of complexity, hormones. Gonadal hormones like estrogen, progesterone, and testosterone hit the frontal cortex hard during adolescence. They influence rates of myelination and the levels of neurotransmitter receptors. This means that milestones in brain and behavioral maturation are less tied to just getting older, and more tied to how long it's been since puberty kicked in. For girls, puberty isn't just about the hormone surge itself, but the erratic way it arrives. Early on, cycles are irregular, and only about half of them actually involve ovulation and a proper hormone surge. So it's not just fluctuating hormones, but fluctuating fluctuations. Boys don't have the same cyclical drama, but there's a humorous, if slightly concerning biological factor. Persistent priapic blood flow to the pelvis during arousal can reduce oxygen supply to the frontal cortex. Hypoxia. It's like the brain's oxygen supply is being diverted, potentially impacting frontal function. Talk about timing. So, let's put it all together. What's the adolescent frontal cortex like? It's a bit of a mess, frankly. You've got leftover synapses that didn't make the pruning cut, communication is sluggish because myelination isn't complete, different subregions aren't quite firing in sync. Plus, the ventral striatum, the reward center, is trying to help out with tasks the frontal cortex isn't ready for yet, but it's like asking a kid to do calculus. And all this is happening while the brain is being bathed in fluctuating waves of sex hormones. No wonder they act adolescent. It's a perfect storm of biological factors conspiring to create that unique developmental period. Despite the chaos, this maturing frontal cortex does bring significant cognitive improvements. Things like working memory, the ability to juggle information in your head, gets better. Flexibility in using rules, organizing complex tasks, and inhibiting impulses also steadily improve. Generally, these gains are reflected in increasing frontal cortex activity during relevant tasks. The more the frontal cortex lights up, the better people tend to perform. There's also progress in mentalization, which is understanding others' perspectives. This isn't just about feeling empathy, but about cognitive perspective taking. For example, teens get better at understanding how an object looks from someone else's viewpoint, not just their own. That improvement in detecting irony mentioned earlier. That's a sign of better abstract cognitive perspective taking, figuring out the intended meaning behind the words. Emotionally, though, it's a different story. Older teens experience emotions more intensely than kids or adults. They're more reactive to emotional phases. Think about the classic amygdala VMPFC interaction. In adults, seeing something emotionally charged activates the amygdala, but then the VMPFC, the emotion regulation hub, kicks in to calm things down. In teens, the VMPFC response is weaker, so the amygdala keeps ramping up. Remember reappraisal, the strategy of reframing situations to regulate emotions? Like seeing a bad grade not as proof of stupidity, but as maybe you didn't study enough. Teens get better at this reappraisal strategy as they mature. Interestingly, early on, reappraisal involves the ventral striatum, the reward system, which is less effective for this task. Later, the prefrontal cortex takes over, leading to more stable emotional regulation. It's a gradual shift towards more mature emotional control. This leads directly into the classic adolescent behavior, risk-taking. They not only take more risks but often seem to have a poorer grasp of the actual odds involved. Sarah Jane Blakemore's work illustrates this beautifully. In one experiment, participants estimated the likelihood of various events, like winning the lottery or dying in a plane crash. They were given feedback about the actual likelihood. Good news like something bad is less likely than you thought, or bad news like something good is less likely than you thought. Then they re-estimated. Adults incorporated both types of feedback. Adolescents? They updated their estimates based on good news, but ignored the bad news. Tell a teen, drunk driving is actually 50% risky, and they might still estimate their personal risk as negligible. This explains why rates of pathological gambling are so much higher in teens. And what fuels this? Dopamine. The reward system, particularly the pathway from the ventral tegmentum to the nucleus accumbens and the frontal cortex, is supercharged during adolescence. This heightened sensitivity to rewards and anticipation of thrills drives that bungee jumping urge. It's not just about risk. Adolescence is also defined by a powerful drive for novelty. Think about how we develop our stable tastes in music, food, fashion during this time, and how openness to new experiences tends to drop off later. This isn't just human. Across rodents, it's the adolescents who are most willing to try novel foods. Among primates, the novelty seeking is particularly strong. In many mammals, adolescents leave their natal group to avoid inbreeding. But look at baboons. A young male sees another troop and slowly transfers allegiance, spending more time there each day until he breaks free. He wasn't pushed out, he was driven by a desperate need to escape monotony. Chimps are similar, with females eager to leave home. The author argues we primates aren't just driven out, we crave novelty. This intense drive for the new seems to be a hallmark of primate adolescence. Let's revisit dopamine in this context. During adolescence, the density and signaling of dopamine pathways, both to the nucleus accumbens and the frontal cortex, steadily increase. Does anticipation release more dopamine? Studies disagree, so absolute levels are less clear than the pattern of release. Consider this fascinating fMRI study. Kids, teens, and adults did a task with monetary rewards of varying sizes. In children, the accumbens response was roughly the same for medium and large rewards. In adults, it scaled proportionally. But look at the adolescents. Medium reward was similar, but a large reward caused an enormous spike in accumbens activity, bigger than in adults. And a small reward? It actually decreased accumbens activity. What does this mean? Adolescents experience huge rewards incredibly intensely, and small rewards feel aversive. Their dopamine system is like a gyroscope spinning wildly out of control. It's no surprise their immature frontal cortex struggles to manage such extreme signals. So, teens have this crazy dopamine system and decent reasoning skills, yet they often ditch logic when peers are around. Lawrence Steinberg's work highlights this perfectly. In a driving game, teens and adults took similar risks alone. But add peers egging them on. Adults stayed the same, but teens tripled their risk-taking. Neuroimaging backs this up. Peer encouragement reduces VMPFC activity and boosts ventral striatal activity in teens, not adults. Why such power? Teens are hypersocial, juggling hundreds of friends, highly sensitive to emotional cues and desperate to belong. This makes them vulnerable to peer pressure, emotional contagion, and deviance training, unfortunately pushing them towards risky behaviors. Eisenberger's cyberball study shows this sensitivity starkly. Excluded teens feel the pain more acutely, with less frontal rationalization than adults. Even at a neural level, watching emotional expressions activates premotor areas linked to imitation more strongly in kids susceptible to peer influence. Conversely, teens resistant to peer influence show stronger striatal responses when reappraising social exclusion. It's a complex interplay of social needs and developing brain systems. How about empathy and moral reasoning? Teens are generally pretty good at perspective taking, saying, "I see how you feel." but they're often better at how would you feel than how does she feel regarding others. Morally, they've moved beyond simple equality to considering merit, but their thinking is still less nuanced than adults. They understand individual circumstances better than systemic ones. They increasingly distinguish between intentional and accidental harm, viewing intent as key. This is reflected neurally, less pain-related activation for accidental harm, and more frontal cortex activation for intentional harm, suggesting appreciating intent is a higher-order frontal task. They also differentiate harm to people versus objects, with the amygdala reacting more strongly to harm to people. Interestingly, punishment for damaging objects becomes less differentiated between intentional and accidental, focusing more on repair. Teens are masters of feeling someone else's pain intensely, sometimes bordering on feeling as the other. This intensity comes from their heightened emotions, openness to novelty, and maybe even a certain adolescent egoism, feeling uniquely empowered to fix things. However, this very intensity can be a double-edged sword. Feeling too much can lead to personal distress, overwhelming the prosocial urge, making them less likely to act. Effective action requires some detachment, the ability to regulate those empathic feelings. Now let's address the darker side. Violence peaks dramatically in late adolescence and early adulthood, then drops off sharply. Think mugger, assault, murder. Interestingly, neuroimaging doesn't reveal a unique brain signature for adolescent violence compared to adult violence. The underlying biology seems similar. Adolescent and adult psychopaths share neural characteristics: reduced frontal and dopamine sensitivity to negative feedback, blunted pain responses, weaker amygdala frontal coupling during moral tasks. And forget blaming testosterone surges. Levels peak early, while violence peaks later. Testosterone doesn't cause violence in adult males either. So, what drives this peak? We'll dig deeper later, but the point is, the average teenager lacks the self-regulation and judgment of an adult. This has real legal consequences. The U.S. Supreme Court recognized this in Roper v. Simmons, banning the execution of offenders under 18, and Miller v. Alabama, banning mandatory life without parole for juveniles, citing precisely this lack of maturity and underdeveloped sense of responsibility. The author agrees with these rulings, but hints that the science discussed here should ultimately push for even broader reforms in the criminal justice system. So why this long wait for the frontal cortex to mature? Is it just that it's the most complex part to build? Probably not. It uses the same basic building blocks as the rest of the brain. The author suggests it's not about complexity, but about evolutionary selection for this delay. Hypothesis 1. Maybe it allows for that crucial adolescent period of exploration, creativity, and risk-taking needed for innovation, like inventing fire or cave paintings. But the counter-argument is strong. For every successful inventor, many more break their necks due to imprudence. Hypothesis 2 feels more compelling. The delay allows the frontal cortex, which handles social intelligence, impulse control, empathy, and self-regulation, skills critical for adult success and happiness, to be profoundly shaped by experience. Think about baboons. High rank comes from muscle, but maintaining it requires social smarts. Adult life is full of complex social forks in the road, demanding nuanced frontal cortex skills honed by lived experience. The irony? The genetic program seems designed to free the frontal cortex from rigid genetic constraints, making it the most experienced sculpted part of our brain, essential for navigating our complex social world. Having journeyed through the turbulent landscape of adolescence, we now return to our starting point, seeking the roots of behavior. Just as we looked at hormones and the menstrual cycle previously, we now turn our gaze back to childhood. Childhood is fundamentally about increasing complexity across all domains, and this complexity unfolds in predictable, universal stages. Research focuses on the sequence of these stages, how experience influences their timing and certainty, and ultimately, how this developmental trajectory shapes the adult we become. Briefly, remember the brain development timeline. Neurons are born and migrate mostly prenatally, followed by a burst of synapse formation. Myelination, wrapping axons in insulation, starts later and proceeds slowly for decades, especially in newer brain regions. This myelination is crucial for connecting distant parts of the brain, enabling functions like toilet training. And as we've emphasized, the later a region matures, the less it's dictated by genes and the more it's shaped by the environment. This sets the stage for understanding how early experiences lay the groundwork for later development.